So, hi everyone. Today, am I audible to everyone though?
Yes.
Yeah. So, today I and Shibani, we're going to present on behalf of our team Neural Navigators on the topic Decoding Neural Circuits for Decision Making, Analysis for Age-Related Differences in Brain Network Dynamics. So, we are a team of five with Zhenrui as a mentor and all of us as members. So let's get started with the topic. So basically, decision making integrates sensory inputs and internal states and adaptive strategies. So hereby we have different states like engaged state, a state and biased state. So in engaged state, we say that decisions primarily are driven from sensory evidence from our environment. For example, we carefully watch the traffic light and road conditions while driving, while in bias state, when decisions show a systematic pattern of error that aren't simply due to not paying attention. Instead, in the state, the error follows a specific pattern that suggests that brain may be using an alternate strategy. So the key insight is that these aren't separate systems, but they are interleaved behavioral modes, meaning that your brain can switch between them or even run them in parallel. So so the primary area of our focus from the studies is the motor cortex, which encodes the variable required for decision making and process the current decision while keeps the alternate strategy in the reserve. And a recent evidence have suggested that aging may disrupt this neural computational capacity, and it can impact the strategy switch and adaptive behavior. So moving on next, for our research focus, we have two questions. First, how do neural dynamic across the secondary motor cortex, basal ganglia, and prefrontal cortex derive the strategy selection during decision making? And secondly, we ask, how does the age-related difference in connectivity among these regions affect the cognitive process and the behavior? So answering these questions, it's significant because we can reveal a mechanism underlying the loss of neural flexibility with age, and uh, that can guide the development of biomarkers and interventions for cognitive decline in diseases like Alzheimer's or any memory disorders like dementia. And yeah. Shivani, next slide. Yeah, so for the data set, we use the Steinmetz data set, which consists of simultaneous recordings from 42 brain regions in mice that were aged between 11 to 46 weeks. And for the data, we process spike data, which was binned into 10 millisecond intervals and normalized, while LFP data were filtered into different frequency bands, theta, beta, and gamma, to extract power features. And all the data were aligned to key task events in the experiments, such as stimulus onset, choice, and outcome. And the sessions were then grouped by the age for comparison. And uh, to decode uh, to decode the neural activity pattern and predict the decision outcomes, we build an LSTM based model. The model take input from all 42 brain regions and begin with masking layer to handle the variable sequence length, and it which is followed by LSTM layer with 64 unit batch normalization and a dense layer, and the outputs are one of the three classes, which is left choice, right choice, or no go. And we train the model using 80 to 20 split with early stopping and Adam optimizer. And we compared the performance to that of SVM, random forest, and gradient boosting classifiers. Now, Shibani will go ahead and talk about the outcomes.
Okay, thank you, Saleha.
So this is the simple LSTM model we built. And let's look at the performance. Actually, before we move to the performance, this is the primary analysis that we did before we started, when we submitted our proposal, before we started the micro publication. So we looked at functional connectivity using the spike data. This was before we started integrating the LFP data. And we did find that there was high connectivity in the between the secondary motor areas and some subregions of the prefrontal cortex. So and this was in line with the literature that we found that regional connectivity strength could predict, better strategy switching capability, and also that these regions were all involved in the decision making time during the duration where the decision making should take place, which is between the stimulus onset and the action that is chosen by the mice. So now when we used our models to predict or rather measure the connectivity and even predict. So first we looked at the connectivity just as a measure, just to measure the connectivity. And we did find that when we looked at different age groups, the younger mice showed more connectivity and a more strength of connectivity as well. And this would show that there was a decline in average connectivity with aging, and this could possibly indicate what we read in the literature, right? That there can be a lowered efficiency and less flexibility in decision making. However, the data that we have doesn't show any reduction in performance. So whether we measure the time or how quickly the mice responded or how successful they were in predicting which way they should turn um, to get the reward, both cohorts, young and old, performed pretty much similarly. There was no significant difference. So that's something interesting we noted. We did assume that possibly this could change if we have more data, but this is, just to be clear, this is what we noted. So even though there was no performance difference, we did note that there were changes in the connectivity. Now, so we thought we could use models to predict this connectivity. And of course, LSTMs performed way better than their counterparts. When I say, of course, I mean because they capture temporal dynamics. So, as you can imagine, decision making is not an event that is, at one snapshot. It's something that goes on for a period of time between when, say, the mice see the stimulus and they make the decision. So, LSTMs are better able to capture such data. So our hypothesis was right, they did perform better. And when we looked at age-specific predictions, we found that the prediction accuracy for older mice was significantly higher than younger mice. We hypothesized from reading up is that um, what we hypothesized is that for older mice, maybe the decision-making processes are more predictable or more consistent. So this could be a reason why a simple LSTM was able to predict it better. Then we also looked at regional contributions. So this is a simple feature importance that we did to see what regions was the LSTM relying more on to predict the accurate results. So what we found was the LSTM showed higher predictive weight on prefrontal regions for younger mice, uh, which is again not consistent with the literature that we found. Literature usually suggests that older brains tend to rely more on the prefrontal cortex and less on specialized areas. But uh, for us, we did find the prefrontal cortex was like a higher contributor for younger mice. Again, this could be also because the LFP data is not explored as much. It's more spike data that we found information about, and also maybe a limitation of the data set being small. But this was an interesting difference that warrants further research. So overall, even though I think our research was a little bit limited due to time, what we did um, conclude on was that we did find strong functional connectivity among these regions, the motor areas and the prefrontal cortex, some subregions, that we did find high connectivity and correlation. Also, again, like I mentioned, there may be a decline, but we did not see any performance impairment. And LSTMs capturing temporal dynamics were our best bet. Like I've said, the small sample size were possibly constrained our ability to generalize. Computational resources, we couldn't leverage the computational resources as much, even though we did have access to some. So that limited our model complexity. And again, our data set only focuses on visual discrimination, while our literature talked a lot about decision making across different types of tasks. So that could be another limitation. So what we would like to do further is have a more diverse sample set, maybe even look at gender differences if possible. Then also more granular inspection of the LFP data. Again, a lot of the research is on the spike data, and we want to explore the LFP data more. And yeah, perhaps more computationally sophisticated models would give us more insights. Thank you everyone. Thank you for the amazing workshops and for our mentor and everyone who helped us through this process. This is our GitHub, which is currently public if anybody would want to give us any feedback or look at it. Thank you so much.